Welcome students. Today we are having a discussion about the unit 1 for the 5th sem students of B Pharmacy, in which the unit 1 is an important chapter about metabolic pathways in higher plants and their determination. It deals with the brief study of basic metabolic pathways and the formation of different secondary metabolites. Through these pathways, we have to study about Shikimic acid pathway, acetate pathways, and amino acid pathways, which comes under the Shikimic acid pathway, and metabolic acid pathway also comes along a branch of an acetate pathway. And the second topic is very, very important, the study of utilization of radioactive isotopes in the investigation of biogenetic studies. The tracer techniques, repeatedly asked questions. So this first chapter plays a major role for the 5th semester student to know about basic chemistry behind the natural sources in the plant and apply your basic knowledge about biochemistry which you studied. And this is also about plant biochemistry and the basic metabolic pathways. So before going to that, let us see what all questions they have asked in Kerala University of Health Science since 2020. 2020 is the first batch. They appeared for the 5th semester exam, in which we can see in January 2021. In 2020, when they kept exam, there was no question have been asked in this chapter. It was surprising that time. But in all the other years, they started asking in this chapter. The question for 2-mark question in January 22, explain competitive feeding technique in tracer technique. We will discuss about this. It's an interesting technique to find out the biosynthetic pathways in plants and the elucidation of the structural elucidation or elucidation of the compound which is responsible for the conversion of the secondary metabolite which is produced from the primary metabolite can be easily identified by means of using this competitive feeding technique, using various radioisotopes. We will discuss about that. Number 2 question, 10-mark question they have asked in November 2021. Outline the acetate pathway and explain its significance. That means what are the products which is produced by acetate pathway in nature plant source of the chemistry. You have to know all the products which is also obtained by acetate pathway. November 2021 they have asked about the 10-mark question. So acetate pathway is very interesting, starting from the acetyl CoA and in between the intermediate product squalene, cholesterol, this all comes in acetate pathway. Even acetate pathway can give rise to the production of terpenes also. Metabolic acid pathway and terpenes, it can give rise to various monoterpenes like limonene, cineole, volatile oil terpene components can also be obtained. In the nature, we can study about the chemistry behind. Number 3, they explain the various tracer technique in the investigation of biogenetic studies. 10-mark question. So we should know in detail about what all the different types of tracer techniques available and how it is performed and how radioisotopes are incorporated and what do you mean by labeled compound and autoradiography and what all the radioactive substances detecting instruments used for the study and what all the significance of the study, what all the research have been done before. These all things have to be explained for this 10 marks. And you see, explain the metabolic acid pathway in the biosynthesis of terpenes. It's also from acetyl-CoA comes to the metabolic acid intermediate product which gives rise to the terpenes, monoterpenes, diterpenes, and it gives rise to the isoprenoid NH and give rise to even vitamin A is also comes under this pathway. So this details 5-mark question in June 2022 they have asked. It gives the birth to terpenes, usually metabolic acid pathway. Number 5, the question in KUHAS they have asked in July 2022. Explain autoradiography. It's a scanning instrument which is in picturizing by means of radioisotopes can be detected by X-rays, by gamma rays. Basic bioanalytical techniques can be determined. It has been explained by many experts, but I have taken some of the points to explain you regarding in this chapter. Explain tracer techniques, 5-mark question. Name 2 basic metabolic pathway in the information or in the formation of plant secondary metabolites. 2-mark question in June 2023. And name different methods of feeding radioisotopes into plant in tracer technique. What are the techniques which can be incorporated the radioisotopes into the plants to have a basic study about the metabolic pathway using tracer techniques? Jan 2023 they have asked by using the radioisotopes. And discuss briefly about the various tracer techniques used in investigation of biogenetic pathway studies. 10-mark question. So you should write all the different types of tracer techniques and how performed and its significance. Applications also should be incorporated. It's 10-mark question, May 2023. Explain in detail the various techniques used in elucidation, characterization, identification of the biogenetic pathways. All the chemical structures, enzymes and the products which are obtained by the precursors is have to be detailed how to do, that is by tracer techniques. 10-mark question repeatedly asked in September 2023. May 2023 also they asked. September 2023 also they have asked. And question number 11, university question, elucidate Shikimic acid pathway, 5-mark question in September 2023. So in same question paper they are asking more than 20-mark questions from this chapter. So it is an important chapter actually. So we have to be prepared for ourselves. This chapter is like a mathematics. Once you read, once you write and study, once you practice for the structures and the chemical constituents or the enzymes responsible for the conversion of the molecules, it will never forget. You can keep it in your mind as a stronghold and no need to have even a time for the revision. You can write the university examination. Explain the acetate pathway and briefly mention the different secondary metabolites formed through this pathway, that is acetate pathway can give rise to various steroids. Even acetate pathway can give rise to various terpenes also. So we have to write about the products which can be obtained. Examples, 10 mark question July 2023. Competitive feeding, 5 mark question in July 2024. So that is also in July 2024, same question paper, July 2024, 10 mark and a 5 mark taken from this chapter. So it is necessary for the students to go through this chapter very detailedly and know about the details, what all the depth of the syllabus they can ask, it's needed. I have taken some of the parts which I can do today, a small lecture only about the Shikimic acid pathway. The aromatic compounds responsible for Shikimic acid pathway in biological study or in biogenetic study or biogenetic pathways. So they may ask specifically Shikimic acid pathway means you should be able to write even Shikimic acid pathway. If it is an essay question, you should write aromatic amino acids like tryptophan, tyrosine, phenylalanine, all are formed from this Shikimic acid. Even phenolic compounds also formed from this Shikimic acid pathway. So that can be written as an example if it is a 10 mark question. So if you need any notes or any support, you can contact me. I will send you by mail or even by the WhatsApp also. It is possible for you to contact me. I can help you. That's why I gave the number and the mail ID, and which can be available in my channel also. If anyone needs the support of the notes in the PDF, I can send to you personally, no problem. So you see, this is the topic, this is the chapter, metabolic pathways in higher plants and their determination. Brief study about basic metabolic pathways and formation of different secondary metabolites through Shikimic acid pathway, acetate pathway, and the Shikimic acid pathway which can responsible for amino acids pathways. It is also written, but it comes from Shikimic acid and conversion of corosmic acid to other aromatic amino acid, and aliphatic amino acids is separately. You should know about that also. And study and utilization of radioactive isotopes, which is used in the investigation of this biogenetic study in tracer techniques. That is a topic which was asked for 10 mark, 5 mark, 2 mark. Individually they are asking about that questions very detailed. So once the students knows about the syllabus and the questions, how they are asking in the university, then if you go for this reading your topics and listening to the lecture, it will be easy for your brain to act according to the examination point of view. So we should know about what is a primary metabolite and secondary metabolite. Before going to a biosynthetic pathway of a plants, you should know basic knowledge about what is a primary metabolite, what is a secondary metabolite. Then only you should study about metabolic pathways. So primary and secondary metabolites, despite the extremely varied character of living organism, plants, animals, fungi, bacteria, so vary in the living organism, the pathway for generally modifying the synthesizing of carbohydrate, protein, fat and nucleic acid are essential, the same of all organism apart from minor variation. Most of them, for the survival, they need carbohydrate, protein, fats, nucleic acid, vitamins even. It's needed for all living organisms. So they vary by plant, that is kingdom of plantae, kingdom of animalia, kingdom of fungi, kingdom of bacteria. But these process demonstrate the fundamental unit, tissues, cells, all living matter, and are collectively described as a primary metabolism, like glycolysis, lipid metabolism. These all are citric acid cycle, Krebs cycle, which is reacting in our body, primary metabolism. Plants photosynthesis, with the compound involved in the pathway, being termed as primary metabolites. The glycolysis, citric acid cycle, photosynthesis, they are all, and lipid metabolism, they are all called as primary metabolism for survival. The products which is obtained from them are products which is responsible for their metabolism. Products which is obtained from them are called as primary metabolites. So primary metabolites are necessary for the basic survival of an organism. This point every student should know. It used for what? Energy and the tissue construction, and include most of them are product like primary metabolism, which all carbohydrates, amino acids and proteins, the building blocks of the tissues and building of our all cells construction, tissue construction, lipids, you know very well, signaling, and the skin, and in many places lipids are more needed for our even circulation structure inside the body also, building blocks. And nucleic acid, DNA, RNA, responsible for various genetic character, and it is necessary, is a primary requirement for a survival of a living organism. And some vitamins also, very, very important. Cofactors. Enzymes and cofactors also very much important for the survival of any living organism. So you see, when you are asked about primary metabolites or primary metabolism, the student should be able to say what is the necessity of the primary metabolism and example for primary metabolites. Secondary metabolites means what? They are secondary. That means are assembled from the pieces of primary metabolites. Assembled, are converted, are biotransformed, are additioned, are multiplied, are made some changes and become into and secondary metabolites, are plentiful and diverse in the plant-based foods, may be more prevalent or unique in certain genus. For example, if you see Solanaceae family, Dathura's family, if you see all of them, they will be having tropane alkaloids. Solanaceae family, so genus, family, so unique, some of them group will be there, species, and similar compounds occurs within genus and maybe families also. Often have vital function in the source. They attract for propagation of species. Propagation of species also done by various predators, various insects from the plant to taken for the pollination. So they attract for pollination, propagation of the species. And you can see defense mechanism predators also some secondary metabolites. They have bad smell, they have sticky nature, which the predators cannot go and attack the particular plant. So defensing mechanism predators also some of the plant will have that secondary metabolite production. Signaling, for various signaling, it can be taken in consideration as a genetic changes and hybrid varieties. So these all are secondary metabolites, and also most of them which is used by human being, and many of them are useful nutrition benefits to humans and other organisms. We are using plant sources as secondary metabolites. So the genes and the enzyme guiding biosynthesis, the genes and the enzymes present in the plants, they will guide the biosynthetic pathways. Biosynthetic pathways vary from one organism to another. So the animal biosynthetic will be different and the plant biosynthetic will be different. And biosynthetic pathway in each plants and the genesis, species will be different by means of presence of their content. So this is a basic picture which will give you explanation about general metabolic pathways, basic metabolic pathways, what we are going to deal with in this chapter. So you can see that clearly. You watch my slide. It shows very simple reaction by the plants. Carbon dioxide and water in the presence of light in green plants, photosynthesis, example they are given in algae, but all plants, it will produce glucose and other carbohydrates. And it can be converted into polysaccharides, you know very well, it will become starch, and even it is cellulose. They all are coming under the polysaccharides. And you see the arrow that carbohydrate can be converted into pentaphosphate pathway, that is a biosynthetic pathway or biotransformation causes to produce nucleosides, RNA, DNA, they are produced, primary metabolite pathway. So you can see again it can also produce erythrose 4-phosphate from the carbohydrate, which is more responsible for us the production of Shikimic acid pathway. The picture, you just see that very clearly, observe. I did not put any arrow, I'm just showing a picture and reading it. You see there, the glucose molecule can undergo even glycolysis. So we have studied in biochemistry, undergoes glycolysis, a glucose molecule can convert it into 2 molecules of pyruvic acid. They are also the same thing, phosphoenol pyruvate. The phosphoenol pyruvate and the erythrose 4-phosphate can give rise to shikimic acid, as shown in the picture very clearly, which is the real pathway of shikimic acid actually. Erythrose 4-phosphate, 4 carbon atom, and a 3 carbon atom containing compound is phosphoenol pyruvate, and that comes to radius steps and gives the shikimic acid pathway of 7 carbon atom containing carbon. And then it can be converted into ligands, coumarins, and it can be converted even to aromatic amino acids, and it can convert it into proteins, enzymes, and even various alkaloids. So you see, the pyruvic acid can be converted into pyruvate, and what is happening? This pyruvate can be converted into acetyl-CoA, then acetyl-CoA, then it enters into mevalonic acid, and that mevalonic acid can be converted into terpenoids, steroids, carotenoids. As I told you, vitamin A. Carotenoids also can be produced from mevalonic acid pathway. And this acetyl-CoA and acetyl-CoA can also produce fatty acids, polyketides, by means of producing cholesterol, by means of producing that structure of steroidal formation, and it goes to produce polyphenols, prostaglandins, macrolytic antibiotics. These all can be produced. And you see again, this acetyl-CoA, we know very well it can be entered into citric acid cycle or TCA cycle or Krebs cycle. This can also give rise to all aconitate, citrate, fumarate, all these structures, or aliphatic amino acids can be produced, which can also give proteins, enzymes, alkaloids which containing aliphatic amino acid, glutamine, glutamic acid. These all can give rise to aliphatic type of amino acids with containing nitrogen. Even tropane alkaloids which contain tropinone. Tropic acid is aromatic. So aliphatic and aromatic mix-up also it is possible for a pathway. So this is just a general idea I want to say. This is a description which I have written is origin of secondary metabolites. 4 major pathway I have written, but only 2 usually. Acetate and shikimate. Acetate can be incorporated with mevalonate. Understand? But you can differentiate in such a way. Acetate, shikimate, mevalonate, and DXP means that is a deoxyxylulose 5-phosphate is also a pathway which can give various structures. So anyhow, the students can write a basic metabolic pathways: acetate, shikimate, mevalonate. Acetate, shikimate, mevalonate. And the key precursors which are used are phosphoenol pyruvate, PEP, phosphoenol pyruvate. Shikimate pathway, 1, acetate-CoA will convert into acetate and or mevalonic pathway. N from amino acids which can added and can give alkaloids. So this is just a short form from the picture which I made it to give an idea. Alkaloids can be formed, phenols can be formed, carotenoids can be formed, proteins can be formed, ligands can be formed, coumarins can be formed, all from the basic photosynthesis initially started by carbon dioxide and water in the presence of light produces carbohydrate. So from primary metabolic pathway, all the secondary metabolites are produced in plant sources. So this makes you to give you a clear picture. Again, I want to repeatedly faster I want to say, listen to this picture and understand the basic metabolic pathway if they ask. You should write this first and then go for Shikimic acid pathway or acetate pathway or mevalonic acid pathway. Understand? Very clearly. So you can write the basic metabolic pathway starts from carbon dioxide in water in the presence of light in the green plants, photosynthesis. It produces glucose and other carbohydrates that can be converted into polysaccharides. And this carbohydrate can enter into the pentose phosphate pathway and gives rise to the nucleosides like RNA, DNA. And also this carbohydrate can give rise to erythrose 4-phosphate. And the glucose molecule can undergo glycolysis and give the 2 molecules of pyruvic acid which can be converted into phosphoenol pyruvate. 1 molecule of phosphoenol pyruvate and 1 molecule of erythrose 4-phosphate combined to give the 7 molecules of structure which is called shikimate. And this shikimate can be converted into lignin or coumarin. And this shikimic acid can enter into give rise to the aromatic amino acids like tryptophan, tyrosine, phenylalanine, and it can give rise to proteins, enzymes, and alkaloids can also be produced as a product. And this pyruvic acid can enter and converted into acetyl-CoA or acetyl-CoA enzyme A. And acetyl coenzyme A can be joined with another acetyl coenzyme and it can give rise to mevalonic acid, and mevalonic acid can give rise to a mevalonic acid pathway which gives the products of terpenoids, steroids, carotenoids. And this acetyl CoA can also be formed squalene, cholesterol, and give rise to fatty acids and polyketides and give rise to polyphenols, prostaglandin, macrolytic antibiotics can also be produced like streptomycin. And you can see it can enter the acetyl CoA or acetoacetyl CoA can also enter into TCA cycle as we have done in the citric acid cycle, which can give rise to various acids like aconitate, fumarate, citrate. These all can be converted into aliphatic amino acids by means of biosynthetic enzymes. So which can give rise to proteins, enzymes, and alkaloids can also be produced, aliphatic amino acids. So this is the main picture of the chapter. So now we should know the metabolic pathway of shikimic acid, which is connected to the aromatic amino acid and phenolic compounds which are produced through the shikimic acid. So we should know what is the shikimic acid. There are 7 enzymes which is responsible to produce the corosmic acid, starting from phosphoenol pyruvate and erythrose 4-phosphate. So these 2 compounds you should remember. So anyhow, we will go for first one. Shikimic acid and shikimate is an important biochemical metabolite in plants and microorganisms. Aromatic compound is a hydrocarbon. What do you mean by aromatic compound? Why you are telling shikimic acid can produce aromatic compound? Aromatic compound means is a hydrocarbon with alternative double bond or conjugate double bond system inside the ring and a single bond between carbon atoms. Benzenes, very simple. Amino acids, the molecule containing amino group, a carboxylic group, and a side chain specific. Phenolic compounds means are large and diverse group of molecules, includes families of aromatic secondary metabolites in plants, various plants like flavonoids. Many are included in these phenolic compounds, even tannins also. Enzymes are group of molecules that serve as a catalyst with a high degree of specificity for a certain substrate or class of substrate. It can only act on one substrate or one on a family of structural similar substrates. So this is just a definition students should know. Major enzymes which are going to deal with, usually in biosynthetic pathway, there are many enzymes, but major enzyme. Synthetase join 2 molecules together without hydrolyzing a phosphatate bond. Dehydratase removes water to create a double bond. Dehydrogenase removes hydrogen atom for its substrate. Removes hydrogen atom for its substrate. Kinase transfers a phosphate group from a higher energy phosphate compound such as ATP adenine triphosphate into a substrate. Shikimic acid, the name from where it came, from this volatile oil, from this compound which you are using, this Shikimi ilicum anisatum. This anisatum, anis, which are used for flavoring agent, which contain the shikimic acid, the structure which I have shown here, with 3 OH and an acid group in a ring structure with 1 double bond inside the ring. Shikimic acid is a precursor for, by means of shikimic acid we can produce, what are the products? Significance for shikimic acid, they can produce aromatic amino acid, phenylalanine and tyrosine, indole and indole derivatives like aromatic amino acid tryptophan, alkaloids, phenylpropanoids, flavonoids, tannins and lignins. They all can be produced by shikimic acid pathway. Formation of shikimic acid. Starting material, as I told, 2. 1 is pyruvic acid or pyruvate, 2 phosphoenol pyruvate. This is a structure. Pyruvate will be converted into phosphoenol pyruvate by converting the ATP to ADP. You are getting phosphatase. Phosphate is connected, phosphoenol pyruvate. And another product which you need is erythrose 4-phosphate. In the first slide itself, I told these 2 products are responsible for shikimic acid pathway. The initial product, by glycolysis you are getting phosphoenol pyruvate, and from carbohydrate production of photosynthesis you will get erythrose 4-phosphate. Erythrose 4-phosphate have 4 carbon atom, and phosphoenol pyruvate is having 3 carbon atom. So when they join together, you will get 7 carbon atom compound. The first step, the first enzyme in this reaction. Always the student should know about what all the enzymes which is responsible for, and the structure's name will automatically come in the enzyme name itself. 3-deoxy, D-arabino, heptilo, that means 7 carbon atom, 3 plus 4, 7 carbon atom compound is going to form. So arabino heptilosinate, 7-phosphate, DAHP, synthetase. Is the enzyme is the first enzyme which is used for the series of metabolism of known as shikimic acid pathway. Since it is the first enzyme, the shikimic acid pathway, it is controlled by how much enzyme is used. According to that, the product will be produced. So it is a controlled amount of carbon entering the pathway. So 7 carbon atom compound is formed by means of this enzyme. 3-deoxy, D-arabino, heptilosinate or heptilosinate, 7-phosphate. You see the structure very clearly. See the structure. Listen to the lecture. You will succeed 100% sure without having any type of reading again and again. Very easy. Erythrose 4-phosphate, 4 carbon atom. Phosphoenol pyruvate, 3 carbon atom, which undergoes this enzyme, that is deoxy, arabinose, heptilose, phosphate synthetase. So that enzyme which is responsible for making a 7 carbon atom compound which is called 3-deoxy, D-arabino, heptilosinate, 7-phosphate. So the enzyme and the product are having similar name. So the second enzyme which you are using is 3-deoxy or dehydroquinate synthetase. So dehydroquinate is formed, very simple, by means of using 3-dehydroquinate synthetase. Is the second enzyme of the shikimic acid pathway. It catalyzes the elimination of phosphate from deoxyarabinoheptylophosphate to generate 3-deoxycunate DHQ. So you see the structure? The 7 carbon atom compound having a phosphate have been removed by this dehydrocunate synthetase, that you can see D-arabinoheptylosate 7-phosphate is converted into 3-dehydrocunate by means of dehydrocunate synthetase enzyme. And there is an NADP, that means nicotinamide adenine dinucleotide, that reduction will takes place by cobalt presence. You can get the third enzyme responsible for our reaction. What is the third enzyme? It is 3-dehydrocunate dehydratase. So the catalyst of the third step of the Shikimic acid pathway, dehydration, removal of water molecule of 3-dehydrocunate to 3-dehydroshikimate. It is converted, dehydrocunate is converted into, getting a double bond by removal of water molecule, you will get 3-dehydroshikimate to the OH and one acid, a ring structure with a double bond OH, simple structure any student can write. Fourth step, Shikimate 5-dehydrogenase is the responsible enzyme. The fourth step of Shikimate pathway is reduction of dehydrogenase synthetase to Shikimate. The reaction is catalyzed by NADP dependent Shikimate dehydrogenase SHD. So dehydrogenase is that is 3-dehydroshikimate is converted into D-shikimate or shikimic acid. The shikimic acid has come, not the completed the pathway, because after corosmic acid, up to corosmic acid we have to write. Shikimic acid is formed. Shikimate means shikimic acid. Understand? Shikimic acid. The fifth step, the shikimate kinase will react now as an enzyme. Fifth step of shikimate pathway. Shikimate kinase catalyzes the specific phosphorylation of the 3-hydro group of D-shikimic acid or shikimate to yield shikimate 3-phosphate. Now you are phosphatizing using ATP as a co-substrate. You can see here shikimic acid or shikimate is converted into shikimate 3-phosphate. Phosphate is getting attached with the OH group. And you can see the sixth enzyme which is used. 5-enol pyrovyl shikimate, because you are adding one pyruvic acid now to this shikimic acid to achieve the corosmic acid. So, enol pyrovyl shikimate 3-phosphate synthetase, EPSPS, is the enzyme you should remember. Enol pyruvic acid is added with the shikimic acid. Just imagine like that, and 3-phosphate is the sixth enzyme of shikimate pathway. It catalyzes the reversible formation of 5-enol pyryl shikimate 3-phosphate EPSP from shikimate 3-phosphate and phosphoenol pyruvate. PEP means phosphoenol pyruvate. So one more molecule of phosphoenol pyruvate is added. So you see here, shikimate 3-phosphate is converted into 5-enol pyryl shikimate 3-phosphate by means of adding one molecule of phosphoenol pyruvate. You can see this structure very clearly. The last step, the seventh enzyme, we are using corosmate synthetase. The seventh final step is the main trunk of the shikimic acid pathway or shikimate pathway, is transferase of 1,4 elimination of the phosphate from EPSP means actually enol pyruvate shikimic acid phosphatase to yield the corosmate is an important reaction. in this reaction, the second of the third double bond of the benzene is introduced. The reaction is catalyzed by the corosmate synthetase. The requires reducing the flavin and activity, even though overall reaction of redox neutral. We need 3 double bonds in a benzene. You are getting this corosmate, the intermediate, 2 double bonds by means of getting this reaction. So enol, pyrovyl, shikimate phosphate is converted into corosmate by means of corosmate synthetase. So that is the enzyme which is giving corosmate. The 7th step of this Shikimic acid pathway links the metabolism of carbohydrate to the biosynthesis of aromatic amino acids and many aromatic secondary metabolites, including tetrahydrofolate and ubiquinone. So there are many aromatic compounds. This is this diagram, or this is the conclusion, this is the summary of what we have discussed now. Just see that 1, 2, 3, 4, 5, 6, 7 enzymes responsible. You can see that phosphoenol pyruvate and erythrose 4-phosphate combine to give a 7-membered 3-dehydro ortho arabino heptulosinate 7-phosphate by means of action of the first enzyme 3-dehydro ortho arabino heptulose 7-phosphate synthetase. And this dehydro ortho arabino heptulosinate phosphate is converted into dehydroquinate by means of the second enzyme 3-dehydroquinate synthetase. And 3-dehydroquinate is converted into 3-dehydroshikimate by means of the third enzyme 3-dehydroquinate dehydratase. And the fourth enzyme shikimate dehydrogenase convert the 3-dehydroshikimate to shikimate, that is shikimic acid, 3OH group and COOH shikimic acid. And the fifth enzyme which is responsible for, that is shikimate kinase, responsible for converting shikimate to shikimate 3-phosphate. And the sixth enzyme which is responsible for converting, that is the enzyme which is used is 5-enol pyruvate shikimate 3-phosphatase synthetase. You are adding 1 phosphoenol pyruvate to get attached with this shikimate 3-phosphate, and you will get the product 5-enol pyryl or pyrovyl shikimate 3-phosphate. And this is again the 7th enzyme which is used is corosmate synthetase, which will give additional double bond in the enol pyruvate shikimate 3-phosphate by means of giving the corosmate structure. So 5-enol pyryl shikimate 3-phosphate is converted to corosmate by means of the 7th enzyme corosmate synthetase. This is the Shikimic acid pathway. Very clearly the short form. Again I am repeating this summary once again. Student, clearly you watch this slide. It is more clear than the previous slide. So the product which you are starting is 2 of them. Phosphoenol pyruvate from glycolysis and erythrose 4-phosphate from photosynthesis. Both the compounds are combined to give a product of 7-membered 3-keto 3-deoxy arabino heptulose 7-phosphate by means of the enzyme reaction 2-keto 3-deoxy D arabinose heptulose 7-phosphate synthetase. And then this 7-membered carb containing compound is converted to 3-dehydroquinate by means of the enzyme dehydroquinate synthetase, that is the second enzyme. And 3-dehydroquinate is converted into 3-dehydroshikimate by the third enzyme 3-dehydrodehydratase. And then the fourth enzyme shikimate dehydrogenase convert the 3-dehydroshikimate to shikimate or shikimic acid. this shikimate or shikimic acid is converted into shikimate 3-phosphate by means of using shikimate kinase. And this shikimate 3-phosphate is converted into 3-enol pyrovyl shikimate 3-phosphate by means of addition of 5-enol pyryl shikimate 3-phosphate synthetase enzyme, which will help for converting this product to achieve, and this product is converted into corosmate by means of the 7th enzyme corosmate synthetase. So this is the 7th enzyme responsible for the shikimic acid pathway. So students have to listen this shikimic acid pathway, write one's structure, and it will be very easy for you to produce in the examination point of view. So this is a small class, only one section I have given. This shikimic acid pathway is responsible for giving various other aromatic compounds like aromatic amino acids, tryptophan, the phenylalanine, tyrosine, and even various phenolic compounds and other terpenoids, all these things, not terpenoids, phenolic compounds and flavonoids, aromatic compounds, coumarins can also be produced by this shikimic acid pathway. Thank you. We will see in the next class.